The small intestine is distinguishable into three regions. A C-shaped duodenum, a long coiled middle portion called the jejunum, and a highly coiled ileum. The opening of the stomach into the duodenum is guarded by the pyloric sphincter. The ileum then opens into the large intestine. The small intestine is highly coiled and is about 5 meters long. It receives secretions from the liver and the pancreas. Besides, its wall also secretes juices. The digested food passes into the blood vessels in the wall of the intestine. This process is called absorption. The inner walls of the small intestine have thousands of finger-like outgrowths called villi. The villi increase the surface area for absorption of the digested food. Villi are supplied with a network of capillaries and a large lymph vessel called the lacteal. The absorbed substances are transported via the blood vessels to different organs of the body, where they're used to build complex substances such as the proteins required by the body. This is called assimilation. In the cells, glucose breaks down with the help of oxygen into carbon dioxide and water, releasing energy. The food that remains undigested and unabsorbed then enters into the large intestine. The large intestine is wider and shorter than the small intestine. It is about 1.5 meters in length. Its function is to absorb water and some salts from the undigested food material. The remaining waste passes into the rectum and remains there as semi-solid feces. The fecal matter is removed through the anus from time to time. This is called egestion. Ingestion, digestion, absorption, assimilation, egestion. The large intestine consists of the cecum, colon, and rectum. The cecum is a small blind sac which hosts some symbiotic microorganisms. A narrow finger-like tubular projection, the vermiform appendix, which is a vestigial organ, arises from the cecum. The appendix was helpful in digesting roughage thousands of years ago when humans used to eat roots and leaves. But now it has lost its significance. The cecum opens into the colon, which is divided into 3 parts, an ascending, a transverse, and a descending part. The descending part opens into the rectum, which opens out through the anus. No significant digestive activity occurs in the large intestine. The functions of the large intestine are the absorption of some water, minerals, and certain drugs, and the secretion of mucus, which helps in adhering the waste particles together and lubricating it for easy passage. The undigested, unabsorbed substances called feces enter into the cecum of the large intestine through the ileocecal valve, which prevents the backflow of the fecal matter. The wall of the alimentary canal from the esophagus to the rectum possesses 4 layers, namely serosa muscularis, submucosa and mucosa. Serosa is the outermost layer and is made up of a thin mesothelium with some connective tissues. Muscularis is formed by smooth muscles. The submucosal layer is formed of loose connective tissues containing nerves, blood and lymph vessels. In the duodenum, glands are also present in the submucosa. The innermost layer lining the lumen of the alimentary canal is the mucosa. This layer forms irregular folds called arrugae in the stomach and small finger-like foldings called villi in the small intestine. Mucosal epithelium has goblet cells which secrete mucus that helps in lubrication. Mucosa also forms glands in the stomach known as gastric glands. The digestive glands associated with the alimentary canal include the salivary glands, the liver and the pancreas. Salivary glands. Saliva is mainly produced by 3 pairs of salivary glands, the parotids located in the cheek, the submaxillary found in the lower jaw, and the sublinguals situated below the tongue. These glands, positioned just outside the buccal cavity, secrete salivary juice into the buccal cavity. The saliva breaks down starch into sugars. Liver. The liver is a reddish-brown gland situated in the upper part of the abdomen on the right side. It is the largest gland in the body. It secretes bile juice that is stored in a sac called the gallbladder. The bile plays an important role in the digestion of fats. It has 2 lobes. The hepatic globules are the structural and functional units of the liver containing hepatic cells. The bile secreted by the hepatic cells passes through the hepatic ducts and is stored and concentrated in a thin muscular sac called the gallbladder. The duct of the gallbladder, known as the cystic duct, along with the hepatic duct from the liver, forms the common bile duct. The bile duct and the pancreatic duct open together into the duodenum as the common hepatopancreatic duct, which is guarded by a sphincter called the sphincter of Oddi. Pancreas. The pancreas is a large, cream-colored gland located just below the stomach. The pancreatic juice acts on carbohydrates and proteins, changing them into simpler forms. The partly digested food then reaches the lower part of the small intestine, where the intestinal juice, or succus entericus, completes the digestion of all components of the food. The pancreas is a compound, both exocrine and endocrine, elongated organ situated between the limbs of the U-shaped duodenum. The exocrine portion secretes an alkaline pancreatic juice containing enzymes, and the endocrine portion secretes hormones, insulin and glucagon. Digestion enzyme action in stomach. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movements of its muscular wall and is called the chyme. The proenzyme, which is the inactive precursor of an enzyme pepsinogen on exposure to hydrochloric acid, gets converted into the active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts proteins into proteoses and peptones or peptides. The mucus and bicarbonates present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated hydrochloric acid. HCl provides the acidic pH, specifically pH 1.8, optimal for pepsins. Renin is a proteolytic enzyme found in the gastric juice of infants, which helps in the digestion of milk proteins. Digestion enzyme action in small intestine. The pancreatic juice contains inactive enzymes, trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases and nucleases. Trypsinogen is activated by an enzyme, enterokinase, secreted by the intestinal mucosa into active trypsin, which in turn activates the other enzymes in the pancreatic juice. The bile released into the duodenum contains bile pigments, such as bilirubin and biliverdin, bile salts, cholesterol and phospholipids, but no enzymes. Bile helps in the emulsification of fats, that is, breaking down of the fats into very small micelles. Bile also activates lipases. Small amounts of lipases are secreted by gastric glands. The intestinal mucosal epithelium has goblet cells which secrete mucus. The secretions of the mucosa, along with the secretions of the goblet cells, constitute the intestinal juice. This juice contains a variety of enzymes like disaccharidases, for example, maltase, dipeptidases, lipases, nucleosidases, and so on. Hormonal control of the secretion of digestive juices is carried out by local hormones produced by the gastric and intestinal mucosa. The mucus, along with the bicarbonates from the pancreas, protects the intestinal mucosa from acid, as well as provides an alkaline medium, pH 7.8, for enzymatic activities. The breakdown of biomacromolecules mentioned above occurs in the duodenum region of the small intestine. The simple substances thus formed are absorbed in the jejunum and ileum regions of the small intestine. The undigested and unabsorbed substances are passed on to the large intestine. Absorption of digested products. Absorption is the process by which the end products of digestion pass through the intestinal mucosa into the blood or lymph. Small amounts of monosaccharides like glucose, amino acids, and some electrolytes like chloride ions are generally absorbed by simple diffusion. The passage of these substances into the blood depends upon the concentration gradients. However, sometimes substances like glucose and amino acids are absorbed with the help of carrier proteins. This mechanism is called facilitated transport. Transport of water depends upon the osmotic gradient. Active transport occurs against the concentration gradient and hence requires energy. Various nutrients like amino acids, monosaccharides like glucose, and electrolytes like sodium ions are absorbed into the blood by this mechanism. Fatty acids and glycerol, being insoluble, cannot be absorbed into the blood. They are first incorporated into small droplets called micelles, which move into the intestinal mucosa. They are reformed into very small protein-coated fat globules called chylomicrons, which are transported into the lymph vessels, or lacteals, in the villi. These lymph vessels ultimately release the absorbed substances into the bloodstream. Absorption of substances takes place in different parts of the alimentary canal, like the mouth, stomach, small intestine, and large intestine. However, maximum absorption occurs in the small intestine.
Know you've been a good friend.
Summary of absorption in different parts of digestive system. Mouth. Certain drugs coming in contact with the mucosa of the mouth and the lower side of the tongue are absorbed into the blood capillaries lining them. Stomach. Absorption of water, simple sugars and alcohol, etc. takes place. Small intestine. It is the principal organ for absorption of nutrients. The digestion is completed here, and the final products of digestion such as glucose, fructose, fatty acids, glycerol and amino acids are absorbed through the mucosa into the bloodstream and lymph. Large intestine, absorption of water, some minerals and drugs takes place. The absorbed substances finally reach the tissues which utilize them for their activities. This process is called assimilation. The digestive wastes, solidified into coherent feces in the rectum, initiate a neural reflex causing an urge or desire for its removal. The egestion of feces to the outside through the anal opening or defecation is a voluntary process and is carried out by a mass peristaltic movement.